Esselamu Aleyküm ve Rahmetullah. Bismillahirrahmanirrahim. I'm Dr. Soylak. I'm from Erciyes University, Kayseri, Turkey. Firstly, I want to start with my best wishes to all of you. I missed all of you. I missed all of my friends at Cairo, all of my colleagues at Cairo, and also I miss all of my friends which attend the conference from foreign countries, including Russia, Uzbekistan, Tajikistan, and others. Inshallah, this pandemic condition will end and we will be together with all of you. I miss you very, very much. And firstly, another special thanks to Dr. Mithat Ibrahim and Dr. Hanan for their invitation. And also, I was not with you last year. I have some pandemic conditions, COVID problem, and I want to thank to Dr. Mithat Ibrahim for his very important support for me about my bad conditions. Alhamdulillah, I am now at better conditions and I can give you a lecture about my studies. I'm working on separation and preconcentration of traces metal ions and traces organic pollutants from the environmental samples. And we are using preconcentration and separation techniques. And nowadays we are using especially on microextraction studies. In my presentation, I will give you a novel. My presentation title is A Novel Approach on the Microextraction and Solid Phase Microextraction for Trace Species from Environmental Samples. And firstly, I want to start solid phase extraction. Solid phase extraction is one of the separation and preconcentration method for the traces analytic ions in the various materials, including natural waters, ores, biological samples, and etc. The main advantages of the solid phase extraction are it is sensitive, fast, economic, large preconcentration factors is possible, simplicity of fast separation, and suitability for automation studies. And in the solid phase extraction studies, a solid phase which we called adsorbent, which has some properties which are given below. It should be porous, its porosity having a large surface area, a material which is a high surface area is it can be used for solid phase extraction studies. Maybe it will be a good adsorbent for trace species, and low blank and high chemical and mechanical stability is very important for the adsorbent because of the especially in the elution step we used and high concentrated acids or bases, and it should be stable for chemicals, and also mechanical stability is very important. Fast kinetics of adsorption and desorption is another important properties for adsorbent. Reversible adsorption, high selectivity and high recovery rates for our analytes is the another important properties for the adsorbent. Nowadays, nanomaterials are usually used for the solid phase extraction studies as adsorbents. Generally, organic adsorbents, inorganic adsorbents, and biological adsorbents could be used. And also hybrid materials, which contains inorganic organic combination, which contains inorganic biological combination, or which contains organic and biological system could be used. For example, as an organic materials, carbon nanotubes, graphene, metal organic frameworks, and as biological sample, we can give bacteria, nanocellulose, nanoorganisms, and others. And for anorganic adsorbents, metal nanotubes, natural zeolites, and others could be used solid phase extraction studies. For example, in this work, we start with carbon nanotubes for the solid phase extraction of traces metal ions in the environmental samples. In this work, we used carbon nanotubes as adsorbent, and we select a ligand ammonium pyridine deuteriocarbamate for the occurrence of metal chelates, or which is adsorption is suitable at the surface of carbon nanotubes, and adsorbent amounts is approximately 300 milligram. We can easily elute adsorbent analytes from the surface of carbon nanotubes by using 1 molar nitric acid in acetone and other conditions. And also, after optimization, all conditions we can easily apply to our methodology for the solid phase extraction of our analytes from food and other environmental samples with satisfactory results. Another example for the usage of carbon nanotubes for solid phase extraction of cadmium, lead, zinc, and nickel ions from food samples by using impregnation procedures with a ligand which is called 4,2,3-azo-resorcinol, and we can easily get quantitative results for our analytes by using only 300 milligram of adsorbent. And then we can apply these procedures for the preconcentration and separation of these ions from food samples, including spinach leaves and other food samples. As pointed in the beginning, we can easily form some nanocomposite materials by the dispersion of particles in a matrix nanometer scale. For example, some nanocomposites are possible: metal ceramic combination, metal combination, inorganic polymer combination, polymer combination, and these nanocomposites are very selective and very good adsorption properties for the solid phase extraction, solid phase microextraction of trace analyte ions. For example, in this paper, in this work, we used polypyrrole and multi-walled carbon nanotubes composite for the solid phase extraction of lead-2 ions in water samples with satisfactory results. This adsorbent has a wonderful, as a good adsorbent for the selective solid phase extraction of lead ions. And also we can use only 1 milliliter of eluate, which is 3 molar nitric acid, and also adsorbent amount is 200 milligram. And also another combination for the solid phase extraction studies, a new hybrid materials which has also magnetic properties. Magnetic properties is very important for us for the selective separation of our adsorbents from the our aqueous samples. Here you can see the collection of adsorbents from the in the both figures. You can see the adsorption of you can see the collection of our adsorbents on the surface of vial by the helping of a magnetic a magnet. And this is very good properties. This is very good properties for us for the separation of our adsorbents from the aqueous samples. And also we applied to this procedure our new adsorbent, which is combination of, which is the hybrid materials with the combination of magnetic alanine amine modified graphene oxide, polyvinyl acetate, covinyl dibenzene composites, which is a new procedure called vortex assisted magnetic solid phase extraction of some metal ions from the environmental samples. Our analysis was lead, copper, cadmium, nickel, and cobalt. And another new application of these procedures by the preconcentration and separation of zeram, which is a pesticide. It was contains zinc ions, and we can easily separate zeram from the aqueous samples and solid phase extraction of zeram by using magnetic carboxylated nanodiamonds, which is a new and good adsorbents for the zeram pesticides. We have applied these procedures to pesticides. Zeram pesticides contains of some food samples and also some water samples. In the preparation step, we are using solvothermal processes for the hybrid materials. If your solvent is water, the procedure is called as hydrothermal synthesis. As you can see in the figure, we can prepare our solid phases, which will be hybrid materials with water. At Teflon pump, we used pressure and temperature at suitable pH, and sometimes, for example, 2 or 3 hours later, new nanostructures will occur. This system is called as hydrothermal synthesis, and we have also a new materials, new composites in our lab for the solid phase extraction of traces organic or inorganic materials. And also we can combine different kinds of materials, including some crystals, polymers, quantum dots, and carbon-based nanomaterials and other materials by using hydrothermal procedure. This is the our hydrothermal processing units. Maybe simply it is a suitable Teflon bump. We can control pH, we can control pressure, and also we can control temperature of this system at the suitable by using, if we use water, this system is called as hydrothermal system. And also it's possible to use different solvents, for example, ethylene, diamine, methanol, ethanol, toluene. You can use different solvents for this Teflon bump system, and methodology is generally called as solvent thermal methodology. And as a sample, I can give some our published works in some good analytical journals. For example, at this work we used molybdenum sulfide and iron oxide nanocomposite as an efficient adsorbent for dispersive solid phase extraction of lead and copper ions from the some water and food samples prior to their determination by using flame atomic absorption spectrometry. This is the procedures. We used ammonium molybdate plus thioacetamide at pH 6, and all the conditions after suitable pH and suitable pressure and suitable temperature, we get these materials. The characterization of these materials is important. As you know, we used scanning electron microscopy at the characterization steps. Also, some mapping procedures for at the D figure are new materials you can see, and or another other characterization techniques, which include Raman spectroscopy, infrared spectroscopy, XRD, and PET methodology. After all of these methodologies, we characterize our new materials, and we can easily use it for solid phase extraction studies. For the all solid phase extraction, solid phase microextraction studies, we optimized all the analytical conditions. For example, as you can see here, the effect of pH is investigated. Also, after optimization, the pH, we optimized the amounts of sorbents, Then we applied to our methodology for the validation of the methodology to certificated reference materials. We can get quantitative results for the certificated results. Then we apply to our methodology to real samples, including water and food samples. Another new materials, which is again hydrothermal synthesis. We obtained it hydrothermal synthesis, and these materials is most like multi-volt carbon nanotubes, bismuth sulfate, nanomaterials for the solid phase extraction of copper. As pointed in the other slides, we get all characterization steps of these materials. After characterization, we used it for the solid phase extraction of copper ions. Another example is nanodiamond plus molybdenum sulfide nanorod composites for the solid phase extraction of lead ions. And we have some review papers about these materials. Also, this is a new one is application of magnetic nanomaterials in bioanalysis, especially for the solid phase extraction of organic traces materials, materials from pharmaceutical, from biological, and from other bioanalysis systems. We have a good review about these materials with my students. Another review about these kinds of works will reviewed in trends in environmental analytical chemistry at this year. This materials gives information about this review gives information about magnetic nanomaterials for the removal, separation, and preconcentration of organic and inorganic pollutants at the trace levels, and their application, their practical application will given this paper. And another one is again important for us, a new, very new paper which is published in Trends in Analytical Chemistry journal, which is very high impact factor with Pınar Gümüş Hanım, metal organic frameworks, which are very important and very high, very good materials for the solid phase extraction of trace metal ions. We reviewed some works on the by using metal organic frameworks in this review. And also we have some works for the preconcentration and separation of some organic analytes, including malation and other organic analytes. This is the by using metal organic frameworks. This is the one of the sample for these kinds of works. Muhammed Habila and Ayman Gahaver is one of my Egyptian students. They are working at King South University. This is our collaborative works with King South University. It was application of metal organic frameworks for the solid phase microextraction of malation from agricultural samples. Malation, as you know, one of the important pesticides in the agriculture. We have a lot of application for these kinds of studies. This one is a special one because this one is two-step preconcentration techniques. At the first step, we have a liquid phase microextraction. Then at the second phase, second stage, we have solid phase microextraction work for the spectrophotometric determination of erythrocene in food and other samples. This was published in Food Chemistry Journal. Switchable hydrophility solvents. I will give some information after these slides about these kinds of solvents, which is a new green solvents. I will give some information about them. Another new application is another new composites, nanocomposites, which contains carbon nanodots plus graphene oxide hybrids for the solid phase extraction of ibuprofen in human blood samples prior to HPLC analysis. This was another application of solid phase macroextraction of organic analytes. And then after this slides, I will give some information about liquid microextraction. In the modern trends of analytical chemistry, today's new microextraction techniques will preferred. These sample preparation steps were selected because of its some of its advantages to use minimum amounts of solvents and reagents, simplicity of steps, high preconcentration factors, green methods for green chemistry, green solvents like switchable solvents like Ionic Liquids, less harmful and harmless than a process with simple step, lower cost, high yield, time saving, low risks, and others. This step is very simple. Generally, we can summarize it like this: addition to extracting solvents at microliter level to sample solution, formation of a cloudy solution, then centrifugation, vortex, and other processes. We can two phases, and our extraction phases is only microliter level. And also after phase separation, we can easily determine our analytes in the extractant phases. At this works, we can use classical solvents, ionic liquids, supramolecular solvents, deep eutectic solvents, switchable solvents, and others. First, we start with classical organic solvents, but everyone knows that these kinds of solvents are very big problem for environment, and we know that their toxicology, the main disadvantages of these kinds of solvents is this. Due to these points, maybe we select other new kinds of solvents, new generation solvents. But at the beginning, we start these kinds of solvents, for example, like chloroform or carbon tetrachloride, but they have some advantages, but their disadvantage, their toxicology, and some of them are prohibited solvents. But I can give an example about these kinds of works: chromium speciation in the water samples by dispersive liquid microextraction combination with total refraction X-ray fluorescence spectroscopy. We used as extractant solvents in this work carbon tetrachloride and chloroform, and we can get quantitative results for the speciation of chromium. And then another kinds of solvents, ionic liquids like organic salts, and they have one cation and one anion sites. And also they are using different at the chemistry with different kinds of parts of chemistry. And we used ionic liquids for the solid phase. We used ionic liquids for the liquid phase microextraction of organic and inorganic analytes. Because of their thermally stable, fast stability is very good, high preconcentration factor we can get, high mass transfer is possible, but the main disadvantage of these materials are expensive. As an example, I can give our work in the published in Desalination Journal, Ionic Liquids Dispersive liquid microextraction of lead ions from environmental samples, and we can get quantitative results by using these ionic liquids for the separation and preconcentration of light ions by using microextraction strategy. Another one is supramolecular solvents. They are combined, they are obtained by mixing, have long carbon chain such as hydroxyl solvents and carboxylic acids with tetrahydrofuran or tetrabutylammonium species, and they are very new and very good solvents, very good green solvents for the microextraction of traces metal ions, because they are environmental friendly solvents. And also fast separation is very simple, and we can get quantitative results by using supramolecular solvents. This is the main methodology. At the end, we can get two different phases. Our extraction phases is the upper level of our wheel. We can easily separate extractant phase to aqueous phase by using micro shrinks and others techniques. And as an example, we can extract to copper in environmental samples by using supramolecular solvent microextraction studies. We applied this methodology for the determination of copper in food and water samples. Another kinds of new generation solvents is deep eutectic solvents. We call them as DES. They are some similar physical properties with ionic liquids. They are very environmental samples. Their price is very lower than ionic liquids, and they have again two parts, mainly chlorine chloride. Vitamin B4 is one part of deep eutectic solvents. Other part is you can select different kinds of materials, and also their most important aspects for having very low freezing point, and they are liquid below 150 centigrade degrees, and also they are most liquid at between room temperature and 70 degrees. And also the application of these kinds of solvents for the microextraction of trace cumin in food and herbal tea samples, as given this procedure, we can get quantitative results for the separation and preconcentration of traces curcumin in food and herbal samples. Lastly, another kinds of new generation solvents is switchable solvents. Switchable solvents have two forms, polar and non-polar form, which they show difference in the terms of their properties. We can easily change the polarity of our solvents in according to our aim. If we need polar solvents, we can use polar kinds of polar. We can convert our solvents to polar form, or if we need a non-polar form, we can easily convert these materials to non-polar form. This is the how we can change the polarity of the solvents. pH is one of the important parameter. We can use pH, or we can use temperature, we can use pressure, we can use carbon dioxide and other. For example, by adding carbon dioxide or by removing carbon dioxide from our solvents, we can easily get which form we need. Due to these points, we use term of switchable solvents for these materials. And I know I don't have too many times, but I'm summarized this methodology. If someone wants to high information, great information about these solvents, they can send email and I can give some additional information about them. This is one of the example which published in Journal of Analytical Atomic Spectroscopy, which is very important journal for Royal Chemical Society. Switchable solvent-based liquid phase microextraction of copper to optimization and application to environmental samples. We optimized all stage of procedures, and we can get quantitative results for the extraction of copper ions from the environmental samples. Then we have some review also about microextraction studies. We published these kinds of reviews in good journals. For example, this one is with my students, one of Pakistani students and one of Turkish students, Özgür Özalp, new trends in extraction of pesticides from environmental and food samples, applying microextraction based green chemistry scenario. This is an important review at these kinds of works. Another one is published at critical review in analytical chemistry, microextraction technique based on new trends especially in food analysis. It is published in critical review in analytical chemistry. Nowadays, we are working on quantum dots in the solid phase extraction, double layered hydroxides in solid phase extraction, covalent organic frameworks at the solid phase extraction studies, new solvents for microextraction, amine solvents and other solvents, nano extraction and new kinds of extraction strategies and solid phase nano extraction, and lastly nano engines and nanomembranes microfactories for the especially drug deliveries and others. And lastly, we have a chapter at this book about microextraction studies, handbook of nanomaterials in analytical chemistry, especially solid phase microextraction and nanohybrid materials. And also we have a book which is published this year published by Elsevier, by New Generation Green Solvents for Separation and Preconcentration of Organic and Inorganic Species. It's published in Elsevier, and also this was a good book at this area, which is written by us. And also I want to thank I'm a member of Turkish Academy of Sciences, TÜBA. I want to thank their financial supports, and also I want to thank the Scientific and Technological Research Council of Turkey, which called TÜBİTAK, and I want to thank my university, Erciyes University, for all supports, and also I want to thank all of you. And I want to some last thanks to my brother, Professor Dr. Mithat Ibrahim, and my sister, Professor Dr. Hannan, and I want to thank all of you. Inshallah, next year we will be together at Cairo, or we will be together at Turkey. Thank you for your attention. Thank you very much for all things.